Thank you, David, for the nice recollection and the nice introduction. Say good day all once again, and welcome to our second and final day of the meeting. We will now hear a series of 4 exciting talks from early career researchers on pressing topics of immunity and vaccines, water, diagnostics, and social science and equity. The format is similar to the one we had yesterday, that is 10 minutes presentation, followed by 5 minutes Q&A for each speaker. First of all, let me stress that when needed, do please then add any question you might have then for our speaker into the Q&A box then throughout the session. Without further ado, let me introduce our first speaker of this session, Dr. Gyaviira Nkurunungi from the MRC UVRI and LSTMH Research Unit in Uganda. He will talk then to us about the effect of intensive treatment for schistosomiasis on vaccine responses among rural Ugandan adolescents. The floor is yours then, Dr. Nkurunungi.
I present this work on behalf of the Population Differences in Vaccine Responses trial team. So we are interested in looking at the effect of intensive treatment for schisto on vaccine responses among rural Ugandan adolescents. A number of studies have shown differences in vaccine responses and vaccine efficacy between high-income countries and low and middle-income countries. And perhaps one of the most studied vaccines is BCG. In this particular trial in Malawi, they showed no benefit of BCG vaccination against pulmonary tuberculosis. Compared to British school children, where studies have shown that efficacy goes up to 70% and above. And a number of other studies have shown that the farther you go away from the equator, the better the efficacy with regard to BCG vaccines. So other vaccines are also affected. This figure is from a study that looked at Ebola virus vaccination between Senegalese and UK participants. And indeed, you do see 7 days and 6 months after vaccination, you do see higher IgG responses among UK compared to Senegalese participants. Within the tropical low-income countries themselves, you also do see differences between different settings. This is from a paper that did work in Gabon, where they looked at flu vaccination and interferon gamma responses in vitro. So 7 days and 14 days after vaccination, you do see that there were differences between the urban and rural setting, differences in interferon gamma responses. We're interested in the differences in environmental exposures that might explain why we see differences in vaccine responses and vaccine efficacy. And our particular interest is in helminth infections because of their immunomodulatory activities in the host. We are currently undertaking a systematic review and meta-analysis. What we see so far is that a number of studies report that you see better responses among the uninfected and those that have been treated compared to those that are infected and untreated. One thing that you also see is that the impact might depend on what vaccine you're looking at. Hence our interest in assessing the impact of parasites, of parasitic helminths on different types of vaccines, on live versus non-live, live oral versus live parenteral, and so on and so forth. In our current work, we hypothesize that Schistosoma mansoni suppresses responses to unrelated vaccines, and that this effect can be reversed, at least in part, by intensive plasquantel treatment intervention. We are working in areas which are endemic for Schistosoma mansoni. These are the Kome Islands of Lake Victoria, which are about 2 hours by powered boat from Entebbe, where we have our labs at the Uganda Virus Research Institute. So we've designed a trial specifically to look at the impact of intensive and helminthic treatment versus standard intervention on responses to a number of vaccines. So we do screen participants, randomize them to intensive, which means treatment with praziquantel at least 3 times before the first vaccination. So these 3 treatments are held each 2 weeks apart, and the last treatment is 2 weeks before the first vaccination is given. In the standard arm, we don't treat until the primary endpoint at week 8. So after the first vaccination, we give 4 weeks later, we give a panel of 4 or 3 vaccines: the yellow fever, oral typhoid, and HPV. And 4 weeks later, we take off samples to do assays looking at primary outcomes. At week 28, we vaccinate with tetanus and diphtheria, and also give an HPV boost. And our secondary endpoint is at week 52, where we also evaluate effect of intervention on response waning. We've collected a number of samples at each of the time points to be able to enable us to look at biological predictors of response to these vaccines. So for the primary trial outcomes, we look at interferon gamma responses at week 8 after BCG vaccination. And for the rest of the vaccines, we look at IgG responses to key vaccine antigens 4 weeks after vaccination. So this is an activity we are currently undertaking. The trial officially ended a number of months ago, and right now we are measuring in the lab these primary outcomes. Of course, I'm still blinded to the trial intervention, so I can only show part of the results. And in this slide, I show that between baseline and the primary endpoint, you indeed do see an increase in response to BCG, to tetanus toxoid, and diphtheria toxoid. For the secondary outcomes, we are looking at proportions with protective antibody and seroconversion rates at 4 weeks post-immunization. So we intend to look at the impact of intervention on these outcomes. Also interested in response waning. So the primary outcome measure is repeated at week 52, hypothesizing here that the infection may accelerate and interventions delay waning. Also interested in effects of intervention on priming versus boosting, and in our case, we can look at this by looking at the HPV responses. We've also used plasma collected at each of the time points to look at Schistosoma mansoni infection status and intensity. In the samples we've so far measured, we see 56% prevalence at baseline. And after intensive intervention at the first day of immunization, we see the prevalence dropping to about 25%. Of course, this is irrespective of trial arm, so we think most of the uninfected here would be in the intensively treated arm. As an immunologist, I'm mainly interested in the exploratory outcomes, where we are looking at the immune response characteristics over the study time course, and also the biological pathways that mediate modulation of vaccine-specific responses. One of the things I'm currently looking at is the pre-vaccination immunological profile through immunophenotyping by mass cytometry and spectral flow cytometry. I'm currently doing this work at the Leiden University Medical Center in collaboration with Professor Maria Yazdembash. We're also interested in looking at markers of gut microbial translocation and associations with helminth infection and vaccine responses. We're interested in the transkingdom concept, where we look at the role of other infections and how they interact with helminths to impact on response to vaccines. Other members of our team are interested in the gut microbiome, transcriptomics, and circulating metabolites, and how these might be associated with parasitic infections and consequently responses to vaccines. As I mentioned, we've already started doing some of the exploratory stuff, and here I'm showing the effect of intervention on markers of gut microbial translocation. We measured responses to a number of markers, and we do see that for all of them, almost all of them, levels or concentrations of these markers are lower in the intensive arm compared to the standard arm. The next step is to, compare these levels in the infected versus uninfected, but also look at associations with responses to the vaccines that we have in our study. So this study is part of the POPVAC program of trials. Of course, for this, we are looking at effect of intensive treatment for schisto. But we've also just started another related study where we're looking at the impact of treatment for malaria on responses to vaccines, the same panel of vaccines. And the third trial is an urban comparison trial whose primary objective is to look at the impact of BCG revaccination on the response to unrelated vaccines. The POPVAC program of trials is mainly aimed at understanding population differences in vaccine responses in Uganda, and we think that this may inform strategies through which vaccine effectiveness can be optimized for the low-income tropical settings where they're especially needed. Thank you for listening to me.
Well, thank you, Dr. Nkurunungi, for this interesting talk. I think it's coming at a time where this much needed control trial of observation between this presence of schistosomiasis and negative then performance in vaccination is being then increasingly then reported. It is also interesting here then to see that this additional burden then of schistosomiasis is being investigated here. So as such, Dr. Nkurunungi, do you think that such an effect is actively mediated by the parasite, or rather come as a result of the host changes, right, in order then to deal with a chronic infection that therefore makes him then less fit to sustain these proper vaccine responses? So more specifically, do you think that such a negative impact will be widely observed for all chronic infection, or is there something particular about schistosoma parasite that mediates this?
Yeah, well, that sounds like a number of questions in one. So the question about whether it's about schistosoma mansoni in particular, or if it's general for all helminths, is that what you meant? If I got you correct?
Yeah, totally. Not just helminths, chronic infections in general.
Yeah. Okay. Yeah. So I mean, yes and no. I'll start with a bit about yes. So chronic infections, if we think of CMV, I think already in literature, there are many reports of prior CMV titers being associated with low responses to vaccines. More recently, talking about the Ebola vaccine studies in Senegal, it's been shown that high CMV titers associated with low Ebola vaccine responses. In terms of helminths, there are a number of studies in the literature, and we see this not just for Schistosoma mansoni, we see it for other, soil-transmitted helminths as well. In Uganda, already in some of our preliminary studies, we showed that Schistosoma mansoni infection is associated with reduced responses to measles vaccine among school children, measles vaccine responses among school children. So I think it's a number of infections. So far, the evidence that's available is for helminths, Schistosoma mansoni and others. But for other infections, it's mainly, I think, in the literature, herpes viruses. You do see inverse associations with responses to vaccines.
Well, thank you very much, Dr. Nkurunungi. Another question then came in on, do you think we could leverage vaccine responses, vaccine programs to distribute helminth treatment in the future?
Yeah,
this is from Anik.
That's a very important question. And one of the things we hope to show at the end of our trial is that maybe national immunization programs should take into account already existing parasitic infections before they do the vaccinations. So in endemic areas, for example, where we are doing our work in the very very schisto-endemic Lake Victoria Islands, we do think it might be beneficial to first do mass treatment in these communities before they roll out certain vaccinations.
Okay. We do have time for one or two more questions. So there are none. I will proceed then to ask any screening that in the
Sorry, I lost you there.
Yeah, I might have some internet problems here. Can you hear me properly now?
Yes.
So did you screen in these cohorts for bystanders infections so as to rule them out? I mean, with relation then to this effect, this negative effect on the vaccine responses?
Yes, we did screen for that. We have samples stored where we are now looking retrospectively at malaria exposure by PCR. We do look at things like HIV and a number of so many other infections. In addition to that, we do have questionnaires that are asked all kinds of questions from other infection exposures to other social and demographic characteristics within these populations. So yeah, we have all that information, and in our analysis, those are things we account for.
Okay, that's great, Dr. Nkurunungi. We do have a question again from Daniela Fusco. She's asking whether you will encourage treatment with praziquantel at the moment of the vaccination in order then to boost the effect. So it will be very interesting then for the pediatric praziquantel strategies.
Yeah, I mean, if our results do show that it's Schistosoma mansoni and this treatment associated with reduced or improved vaccine responses after treatment, I think that would be the next outcome to kind of advise policymakers to treat for parasites such as schisto before rolling out vaccinations in such communities.
Thank you, Dr. Nkurunungi. We do look forward into the outcome of this trial. Me particularly, actually, as we do have a similar study engaged in here in Cameroon. So thanks very much again for sharing that with us today. And we will now go to the second talk of this session from Dr. Laura Braun from Imperial College London, who will present then on the chlorination of Schistosoma mansoni cercariae. Dr. Braun, then the floor is yours.
Hi, my name is Laura Braun, and today I will be presenting about the effectiveness of chlorine against schistosome cercariae. This was research that I conducted as part of the WISER project, which was a collaboration between institutions in the UK, Ethiopia, as well as Tanzania. One of the objectives of the project was to determine how effective water treatment processes are in killing schistosome cercariae, and we focused on 3 treatment methods that are all considered suitable for low resource settings. But today I will only be focusing on chlorination. Looking at the life cycle, we can identify the main ways of cutting transmission, which include drug administration, sanitation to contain the eggs, as well as snail control. To reduce infection, one has to reduce the contact with water that contains cercariae. And this is hugely challenging because communities very often depend on freshwater bodies for household, recreational, or occupational activities. One potential solution is providing communities with treated water. Water can be abstracted from a freshwater body and then treated on a household or community scale, where it can then be used in water infrastructure, such as laundry stations, shower, or recreational areas. And by treating the water, we don't just provide communities with clean water, but we can also potentially reduce the time that they spend at transmission sites. When I started my PhD, I tried to answer the question, how do you produce safe, cercariae-free water? So essentially, if you collect a bucket of water from a transmission site, how do you make that water safe? And I first looked at recommendations, for example, by the WHO, but these only stated that you should boil water for 1 minute, which is not necessarily practical. So we then tried to answer this question by conducting a systematic review. Without going into too much detail, the results essentially showed that there was huge variability for all the treatment methods. And we concluded that there were both insufficient and incoherent data to develop guidelines. And because of this, we decided to go ahead and conduct water treatment experiments. Our experiment used Schistosoma mansoni cercariae, and we produced these by extracting eggs from infected mouse livers. We then hatched these eggs into myristidia and exposed these to snails that were bred at the Natural History Museum. And after about 3 weeks, the snails then started to produce cercariae. Most of our experiments were run at the Natural History Museum using these lab-grown cercariae, but we also replicated experiments in Tanzania using snails collected from a transmission site. And this was to see if our lab-grown cercariae and the wild cercariae were equally sensitive to chlorine. The experiments were relatively simple. We had a beaker of water with 50 ml of water at 2 temperature and 2 pH conditions. We added 100 cercariae, dosed the chlorine, and ran experiments for up to 45 minutes in 5-minute increments. And at these 5 increments, we would assess how many cercariae are alive and dead by counting the number of moving and non-moving cercariae. So a non-moving cercariae was assumed to be dead. And we then measured the residual chlorine using the titration method. I'm sure you all know what cercariae look like, but in case you haven't seen one for a while under a microscope, this is what they look like, and they are visible to the eye. In this video, you can see what it looks like when cercariae are inactivated. So at first, the cercariae swim quite freely, but then as the contact time continues, the cercariae start to sink to the bottom of the beaker. They become quite sluggish, and we consider them to be dead or inactivated when they are completely non-moving, as shown here. These 2 graphs show what CT values are required to inactivate cercariae. And so a CT value is the product of the chlorine that's available for disinfection and the contact time. So essentially a CT value is a measure for how long an organism has been exposed to a certain concentration of chlorine. The first graph on the left is for 20 degrees, and the second for water at 27 degrees. And the 3 lines represent the 3 different pHs. There are 2 main findings from this research. The first is that the CT values at 27 degrees, this graph, are lower than that at 20 degrees. And that is expected because chemical reactions are faster at higher temperature. And the second observation is that higher CTs are required at higher pH, which you can very clearly see in this graph. And this is because under alkaline conditions, chlorination is less effective. We then use these graphs to read off the CT values at given log inactivations, and thereby produce what we, in water treatment, we call a CT table. So here you can see the differences in CT value for the different pH and temperatures more clearly. And we read off the highest, most conservative CT value of 26 milligram minutes per liter. So far, this has all been for laboratory-based experiments. But we did also replicate these experiments in Tanzania using water and snails that were collected from this transmission site by Lake Victoria. And unlike before, we did not control the temperature and pH. Now, although we can't directly compare the results to our previous graph, because the pH and the temperature aren't exactly the same, you can see that the field results, which are shown in blue here, are in line with our lab results, which we confirmed with statistical analysis. And this means that our lab results are also valid under real-world conditions. We also repeated these experiments for aged cercariae, which were approximately 6 to 9 hours old, compared to fresh cercariae, which were always less than 2.5 hours. And in the graph, you can see that the aged cercariae in the dotted line here, or dashed line, required much lower CT values than fresh cercariae. And again, this is confirmed by statistical analysis. In case you're wondering why the cercariae are inactivated by chlorine, this shows one of the mechanisms, which is that chlorine actually ruptures the surface membrane of the cercariae head, shown by all these holes. So our raw results under worst case conditions showed a CT of 26 milligram minutes per liter to achieve a 2 log inactivation. So 2 log is equivalent to 99% inactivation. We then apply a measure of uncertainty, which brings our recommended CT value to 30 milligram minutes per liter. And this could be achieved, for example, through a chlorine residual of 1 milligram per liter after 30 minutes of contact time. And it's this value, the CT of 30, that will be published in the Schistosomiasis fact sheet by the WHO. Comparing the disinfection to other microorganisms, you can see that Schistosoma mansoni cercariae are more resistant than E. coli, for example, but slightly more sensitive than cholera. So in conclusion, we have tested the effectiveness of chlorine against Schistosoma mansoni cercariae and found that they are relatively sensitive to chlorine, requiring a CT of 30 milligram minutes per liter. And that is valid for water that's at least 20 degrees and less than a pH of 7.5. This research has answered some of the technical questions on how to inactivate cercariae using chlorine, but the challenge will definitely be finding ways to implementing this sustainably in communities, for example, through laundry stations or recreational water areas. Whatever the solution, it will certainly have to be very context specific and should ideally be developed alongside disease and wash programs with input from the community. I certainly don't see water treatment as being a standalone control measure, but rather that it gets integrated into existing control programs, because that way the overall disease burden is reduced through drug administration and reinfection is potentially slowed due to reduced contact with cercariae contaminated water. I'd like to thank the entire WISER team, especially Jacinta, Mullework, and Meseret, who helped lead some of this research. Thank you very much.
Well, thank you, Dr. Braun, for this insightful talk. We do have a couple of questions, starting from Jean-Pierre Mukendi, who is asking, what water did you use in the beaker for this beaker experiment? Was it the deionized water or the tap water?
Yeah, thanks for that question. So when we ran experiments in the lab, the Natural History Museum, we used bottled water. And the reason for that is that we wanted to control the pH without adding buffers. So we had 3 types of bottled, unchlorinated bottled water that were at pH 6.5, 7, and 7.5. And when we then replicated the experiments in Tanzania, we used natural water, untreated natural water from a transmission site. Yeah, I do think that if you would use distilled water, that would actually kill the cercariae after some time.
Well, thank you. Carlos Texera is asking, for how long do you look for the non-moving cercariae to say they are dead? Yeah,
that's a bit tricky. So we generally looked at them, I would say on average for about 5 seconds each. We do look at, we had 100 cercariae per beaker, and we didn't want to spend too much time looking at each one, because that would mean that your experiment would take quite a long time and you'd introduce some uncertainty that way. But we did actually poke the cercariae with a needle as well, just to make sure that they wouldn't react. Or well, if they did react, that would mean that they were not actually dead. So that was a kind of way of doing a visual test, but also a physical test.
Okay. And we do have a question from Christophe Greveldin asking whether the cercariae can recover from this chlorine stress. So when the solution is exchanged again against water afterwards, is it possible for them then to recover? In other terms, then, is this chlorine effect irreversible?
Yeah, no, that's also a really good question. It is not reversible. So in one of the last images I showed you, the cercariae are actually physically damaged by the chlorine. And so once they're chlorinated and non-moving, that actually means that they are inactivated and they are dead. And we tested that by diluting the water. So essentially reducing the chlorine concentration to close to zero to confirm that they actually would not start moving again. So yeah, once they're chlorinated, they are completely inactivated.
And Zygmunt Bartonisek then asks, says, first of all, hi Laura, great presentation. So how does the CT value you have recorded compare to value obtained when using, for example, chlorine tablet or standard chlorination of drinking water?
So the form of chlorine shouldn't matter too much. The most commonly used chlorine form is sodium hypochlorite. If you then start to use, for example, chlorine gas, the CT value might change a little bit, but generally the tablets also contain sodium hypochlorite. So the CT value would not change depending on what form of chlorine you use necessarily. And what was the other part of the question?
The other part was how does that then relate to the ones that are currently used or generally used then for water?
For drinking, okay.
For drinking water.
For drinking water. So generally, the CT value for drinking water is around 30 milligram minutes per liter. So it's actually exactly the same as we found in our results, which is really promising or really positive, because that means that in theory, the water that treats cercariae can also be used for safe drinking water.
And then Jacqueline Orford then is building on that to ask whether you will need now more safety factors if you'll be using this in the field. So more than the 4 milligram per liter that you've added.
Yeah, so that 4 milligram minutes per liter of safety factor came primarily from uncertainty in equipment as well as experimental errors. I don't necessarily, well, I don't want to say that you shouldn't add safety factors, but we don't think that you would necessarily need an additional safety factors, because first of all, our experiments were completely based on 100% fresh cercariae. And as you saw in the later slide where we tested aged cercariae are actually a lot more sensitive to chlorine. So by assuming that you have 100% fresh cercariae, you're already building in a safety factor. And the other thing is that we used quite high cercariae concentrations. We used 100 cercariae per 50 milliliters, which is, I believe, 2,000 cercariae per liter, which again, you would not necessarily find in the natural environment unless you've scooped up a snail in your jerry can. So for those reasons, I think the safety factors that we've built in, bringing in up to 30 milligram minutes per liter are sufficient.
Okay, we do have a couple of other questions that will compile into one final question, which is basically regarding how safe this practice will be then for the environment, regarding the ecosystem and stability, and how this then will be perceived then at the community level. So,
I mean, in terms of the environment, we're actually not adding the chlorine to the lakes or to the freshwater body, but this water would be abstracted and then treated either on a household or community scale. So we're actually not affecting the environment at all that way. And chlorine is relatively safe to use, especially at these levels. It's far below the health thresholds that are recommended by the WHO. So yeah, there should be no environmental or health concerns with these levels.
And community acceptability gives
Sorry.
regarding the community acceptability of using the chlorine. Can you hear me?
Yes, I can now. Sorry, I think my video was frozen for a little. I mean, chlorine is one of the most commonly used forms of disinfection, and it's very well known and in general very well accepted. There are issues, of course, with taste and smell, but because this is a relatively low level of chlorine, and you know, if because we're talking about schistosomiasis, the primary focus is not drinking water, but more water for household or recreational activities. So there hopefully shouldn't be too many barriers in the acceptability, but of course this will need to be looked into and researched in future studies.
Well, thank you, Laura. And please do check on the Q&A for more questions that are coming in that you might be able then to answer directly there. And thanks very much then for the interesting talk.
Yeah,
so
great.
without further ado, we will move then to the next talk from Dr. Anna Kildemoes from the Department of Parasitology at Leiden University Medical Center in the Netherlands. And she will talk to us about towards accurate schistosomiasis serology using a Glycan microarray approach. The floor is yours, Anna.
Hello everyone, my name is Anna Kildemoes and I'm part of the Leiden Parasite Glycobiology group, which is led by Professor Ron Hocke. First of all, I'd really like to thank the GSA organizers for allowing me to share some of our work with you today. And what I'm going to talk about is how we use Glycan microarrays to identify schistosome antigens with diagnostic potential. And that is, of course, with the aim of future development of an antibody-based diagnostic tool. So it's very well known that schistosome infection elicits a lot of antibodies, but it's less well characterized what these antibodies actually bind to. We do know that many of the parasite antigens are glycosylated, and it could be glycolipids and glycoproteins, and that many of these are antigenic. So in the team, through extensive glycomics work, we've mapped basically glycan elements that are unique to schisto compared to humans, and those are the ones we are interested in. So that maybe we can identify a single or a combination of a few that we could combine into a very specific and very sensitive tool, basically exploiting the immune system amplifying signal to very little antigen. So if we manage to develop such a tool, then the use case for it would be in endemic area context, very low or near and post-elimination monitoring, as an antibody-based diagnostic tool can't distinguish current and prior infection, but it can be very useful in terms of monitoring transmission, for example, in young children, in occupational risk groups, and also in hybrid and zoonotic contexts, it might have added value. So in non-endemic contexts, it also could be very useful for traveler diagnostics. And if we manage to eradicate schisto from some areas, it would also be useful for monitoring there, as well as in emerging risk zones mediated by climate change. Okay, so if we imagine diagnostic scenarios for antibody detection, then basically we have one where a person is naive and have no antibodies towards schisto. We have one where it's a primary infection and they may or may not have any X, potentially also quite a low dose exposure. We have a current and chronic infection where a person may have been exposed and reinfected multiple times, and probably also people have quite varying intensities of infection. And then of course we also have a prior exposure infection where either a person is successfully treated or worms have simply died of old age. So it's very possible that these different scenarios have different antibody profiles, both in terms of isotypes and subclasses, and also which antigens they actually recognize. So we are trying to map this and then use that knowledge to identify diagnostic candidates. So the technique we use to investigate that is the Glycan microarray, and basically we make custom arrays with the relevant molecules from parasites, and we can print those in nanodrops on glass slides and covalently link them and immobilize them using this microarray printer. And then basically what we do is make a mini ELISA on the slide. So we add sample, and antibodies in the sample bind to the targets they recognize. And then we use secondary antibodies with fluorophores. We can use 2 fluorophores at a time. to detect antibodies that are bound to specific targets. So for example, here we have a gasket where you can have 8 samples, and within each cavity there's an entire array. In an array, you can see here an example of a person having a lot of antibody to this triplicate, but nothing to the triplicate next to it. So basically we get a profile of a person's antibody response. So each bar here represents a target. And we then use well-characterized samples to find out which targets could be useful in a diagnostic context. And we consider several isotypes and subclasses, and of course also cross-reactivity. To identify targets of interest, we used an iterative process where we first started with a large array and defined smaller sample sets. We then upscaled this to validate the first results, and we revalidated in secondary sample sets. And we also ran a lot of samples of true negative samples to ensure that we are looking for both sensitivity and specificity characteristics for certain targets. And I'm going to show you results for one marker that looks promising for acute schistosomiasis. So first of all, we have access to controlled human infection material via Meta Rustenberg and the COSI team. So these are 17 healthy volunteers with male cercarial exposure of 10, 20, or 30 parasites. And basically what you see here are time courses over weeks post-infection for, on the left-hand side, IgM responses and the right-hand side, IgG responses. And very defined material like this is really valuable because we learn, first of all, about specificity. We select the targets that are truly negative at baseline. And we look also at the sensitivity, that we need all the people infected to become positive for certain targets. And then we also learn a lot about when a test actually can be used for certain candidate antigens. So when are antibodies consistently raised in people to the antigens, and how long do these antibodies actually persist? That gives us a lot of clues to how to apply a diagnostic test. So this looks really good, but these are only 17 people. So just to show you some data on specificity. So here are some of our results from our upscaled specificity validation. So true negative samples. On the top are IgG results, and on the bottom IgM results. First, it's the COSI samples, so the controlled human infection samples. Then we have some donor samples from the Netherlands blood donor bank. And we also have travel samples from the Netherlands. So these are baseline samples of a pre-post travel study that was collected between 2007 and 2009. And we also have access to samples from 2009 from Flores Island via the Immunospin study. And these come from a salt-transmitted helminth endemic area, quite high transmission. However, on this island, there should be no schistosomiasis. So overall, these are the specificities for all studies pooled together. You can see that the Flores Island perform a little bit less good. However, over 85% is still a good performance. It cannot be excluded that some of these individuals have traveled to nearby islands where schistosomiasis is endemic, and hence they could have been exposed. So we aim to run more samples to validate the specificity results. Now, to illustrate sensitivity in travelers here, I'll just show you the paired results from the Dutch pre-post travel study. Originally, there were 9 seroconverters detected. We pick up 6 out of 9 on IgD in array, and we pick up 9 out of 9 for IgM on array. We actually also find 2 additional positives for IgM. I do not think these are false positives, but rather that the array is very sensitive, so they are not picked up in the original study. We also, very promising, have a new sample set via IS Global in Barcelona and José Muñoz. And these were 6 young men who went to Mozambique and got subjected to schistosomiasis at a single exposure, a mansoni clade parasite. And for those, we have very, very high antibody titers towards our antigen, both for IgG and IgM. So these are very promising sensitivities, I'd say. So to sum up, I hope I've convinced you that we've identified an antigen which looks promising, particularly for acute or primary infection schistosomiasis. We've also gained a lot of knowledge on antibody profiles in terms of which isotypes and also subclasses are present specific for certain antigens. And importantly, we've also learned about the longevity of responses to those antigens, which is all information that can help us in defining our use case and actually apply a test once it is eventually made. What we need to do is, and what we will do in the coming months, is upscale validation in terms of cross-reactivity to other infections. We also have a lot of data from endemic area samples. Those are a bit more complicated to interpret as we're dealing with both current and prior infection in the mix. So that takes really careful analysis. What we hope to do in the future is move towards the technical feasibility stage for actual development of a test, such as a finger prick test, a rapid test, for example, in a lateral flow format, as this Glycan microarray platform really is not a diagnostic tool, but is for identification of the targets. And we hope that you agree that an antibody-based diagnostic tool could be useful for the scenarios I introduced to you in the beginning. So a huge thank you to international and local collaborators, as well as LMC colleagues. Special thank you to study participants and clinical and fieldwork team members. This work was done under the Detect Schisto project, which is a collaborative consortium between Nagasaki University, LOMC, and Ligature as project management partner, also closely collaborating with KEMRI in Kenya, and we were funded by the GHEAD Foundation. And finally, thank you all for your attention, and questions are very welcomed.
Well, thank you very much, Dr. Kildemoes, for this interesting talk. And we do have a couple then of questions that are pouring in, but this I must first of all point out is pretty much timely at the time where we'll look for the most sensitive then tools to be able then to proceed with impact assessment. So this is very timely and a wonderful job that was done there. Congratulations, Anna. So we have a question from Carlos, from Eric Locker. So setting that, is there any indication that people who have had swimmer's itch due to avian schistosomes will test positive?
So thank you for that question. That's actually a really good point, as many of these epitopes are indeed glycans. So we have other work where we've just tested Trichobilharzia regenti cercariae. Of course, there's several bird schistosome species. So we actually just had a student coming in with results looking at cross-reactivity for that. For some epitopes, it is there, but it's not very high. So I'm sure we can, through technical advancements, get around that. But it's definitely a point.
Okay. We have Christina Fonseca that is asking whether the response of these individuals, for example, in control infections after treatment, how was it? So did you get to evaluate that?
Yeah, they were treated at 12 weeks post-treatment, and then a couple of people needed additional treatments. You can find that in the Langenberg paper from Meta's group. But basically, the antibodies stay high, and particularly the IgM response stayed high even a year post-infection. So that was a bit surprising. Now, it has to be said that that this area is very, very sensitive. So, so perhaps with other diagnostics tests, you can catch dynamics a bit earlier, but you definitely have circulating antibodies for a long time.
And Elias Hinango is asking whether with samples from endemic setting, would you expect then a correlation between these antigens and the IgM levels? So I think
well, maybe not correlation, but association, I hope. It's a bit difficult with doses. We actually do see a dose response in terms of initial exposure dose in naive, so in primary infections. But in endemic areas, the question is the dynamics between a primary infection and then if you have a boost effect. And I actually suspect after many years of exposure that you get a downregulation to certain epitopes again. So we need more knowledge about in which age group and under which transmission pressures we can apply it. But we have a lot of data coming in, so I hope to be able to answer some of that quite soon.
Great. We are running out of time, but equations that I also had in mind from Uwe Mekpo asking whether you can measure intensity actually with this test.
Yeah, I think you perhaps can get an indication of that in primary infections. I don't think you can in chronic infections. It really is an exposure marker, but you could maybe think of it as a screening tool where you can narrow down if there's a problem in an area, and you can then go in with a parasite specific, such as either an RT-PCR or other molecular diagnostics, or an antigen tool to then confirm the infection in individuals and choose your treatment strategy. So maybe saving money.
Well, quite a lot of questions that are pouring in. So Anna, please, will you get into the question and answer section to try and answer these? And without any further ado, first of all, thank you very much for the talk once again. And without further ado, we'll get now to the fourth and last talk of this session, which is on an equally, if not a more important aspect of the schistosomiasis than research. That will be by Mrs. Christine Masong from the Catholic University of Central Africa in Cameroon, who will present on achieving equity in UHC and interventions, basically who is left behind by neglected tropical disease programs in Cameroon. The floor is yours then, Christine.
Hello, everybody. I am Christine Makia from Cameroon, and I'll be presenting to you a summary of our paper on the social science perspective on equity related to achieving equity in universal health coverage interventions. Who is left behind by neglected tropical disease programs in Cameroon? This is based on a case study of mass drug administration for schistosomiasis. So this paper came as a result of a qualitative study embedded in the Countdown Consortium's multidisciplinary research in 4 regions of Cameroon. And our objective here was to assess which groups are constantly missed during mass drug administrations and developing strategies to enhance equity. So for this study, we used an ethnographic process where we met participants from all levels of the community as well as the healthcare delivery system. And through this ethnographic process, we used participant observation, in-depth interviews, and focus group discussions across all of this population in order to throw light on the existing equity challenges. So as I mentioned, we had people from the grassroots, people from the intermediate level, and also at strategic level. So at the grassroots, we met community members made up of parents who had children in school who had either benefited from mass drug administrations or had experienced mass drug administrations in the community. We had teachers as well who were involved in school-based distribution of praziquantel. We had community drug distributors within these communities as well as village leaders. At the level of the intermediate level, we had wash partners involved in supplying and supporting with infrastructure as well as wash services. We also had government councils, local councils and government councils who were involved in hygiene and sanitation activities as well as providing wash infrastructure. And we also met health officials. At the level of the health area, we had the chief of center, we had the district medical officer at the district level, we had the regional officer, and we met some NTD coordinators and focal persons. At the strategic level, at the central level, we came in contact with technical partners and funding bodies involved in the fight against neglected tropical diseases in Cameroon. So I think it's important for us to insist why we met so many people at the level of the community. This is because school-aged children, 5 to 14, fishermen and farmers are known to be high-risk groups for schistosomiasis infection, as well as women and girls, because the gender roles mean their daily activities include a lot of freshwater contact. And so that was the reason why we met these people in the community as well. as I mentioned, we worked in 4 regions in Cameroon. So you can see on the Cameroon map, where highlighted in green, you find Malantwain Health District, Edea Health District, Monge Health District, and the Kumba Health District. And in these health districts, there had been carried out both school-based and community-based mass drug administrations for schistosomiasis as part of the countdown program. And so we had a total of 109 in-depth interviews with 6 focus group discussions, all drawn purposefully from the community. So we received ethical approval from the Cameroon National Ethical Committee on Human Health Research. So the participants in the collection of our data, what we focused mainly on was asking participants to reflect on previous mass drug administration based in schools and communities. And also I want to point out here that data was collected at different times, different points during mass drug administrations. We had data collected up to 1 month after the last drug administration, and data collected during the period of mass drug administration. So we transcribed our data and translated into English from Pidgin and French. We coded and set for themes, and finally we generated themes using a thematic map. So our results show, from our analysis, 4 groups of people were mostly being repeatedly missed in mass drug administration campaigns, and these were the indigenous farming communities, migrant farming communities, out of school children and school children in private schools, as well as young girls and women of reproductive age. So the indigenous farmers were the first settlers in these communities. And we found that they were mostly missed because they were away working in their farms during mass drug administration campaigns. And this was due to an insufficient or ineffective sensitization from the community drug distributors and the other stakeholders who carried this out, and also because of the sensitization which was given, the insufficient sensitization, there were existing myths that continued in the communities and which limited people from participating, such as fear for economic loss due to inability to work after consuming parasitic. Also, we had migrant farming communities which are predominantly Nigerian or migrants from other regions of Cameroon, and who were missed, but particularly because there was a lack of engagement of existing networks. They had trusted networks which were not met during these campaigns, and these people were not met because they didn't trust what was being brought to them. Also, we had out-of-school children and children in private schools, which were missed because of existing strategies were not capable in meeting them at all times. Also, young girls and women of reproductive age were missed due to pregnancy or lactating, where most times when these campaigns were carried out, either these women were not given treatment because of unclear policies or unclear strategies in giving parasitic to pregnant women, and also there existed myths around fertility and parasitic, which limited these women from receiving treatment. So these findings from our results which I presented have shown there exist gaps which have created circumstances where people or groups of people are repeatedly missed in MDAs in Cameroon. And this, as we said, is due to inadequate or insufficient sensitization campaigns, also limits in CDD training capacity around distribution of drugs to pregnant women, and also the method of sensitization. Also, there lacks alignment between distribution and availability of many community groups, as well as the one-size-fits-all approach is not tailored to the realities and challenges of specific groups, and this results in them being missed. So in this, we confirm in this paper the importance of understanding the challenges faced by marginalized groups in designing MDA campaigns, and also there's a need to break down populations into different groups in order to meet on with context-specific axes. And also we conclude in saying it cannot be overemphasized in the need in embracing formal and informal social networks which exist in these communities to promote more equitable MDA. So with the evidence presented here, NCD actors could and can be able to take steps to understand the gaps and challenges needed to reach these at-risk populations in order to meet schistosomiasis elimination goals. Thank you very much, but this paper was not just for myself, but we had a host of other authors, as you can see here, and I encourage you to find time to read this article. Thank you very much for your time.
Well, thank you very much for that presentation. This is the Masong. Are there any question on the chat? So while waiting for those then to be typed, I see quite a lot of them coming. First of all, Carlos Texera asking or stating, first of all, that this type of study is critical, where people trust more from the community than health agent from the government. So have you noticed that in your study?
Oh, thank you very much. Actually, that was one of the main things in our study. When we talk about trusted communities, trusted leaders within the communities, this was what we meant. Because we met whole groups of people within communities who were not willing to listen to the distributors because they trusted another set of people who were not involved in mass drug administrations. So I think from what we understood from the communities was what was the main problem in most within communities. Networks were not considered when treatment interventions were going on. And this was really actually one of the main gaps we experienced. Thank you.
So we do have another question from, I think the pseudo, Mivian, stating that will the age not have an impact on the study? The age of the participant, I think they meant.
Excuse.
Did you get me?
Actually, this is a paper I invite you all to read. Yes, I did. Thank you. So the age actually, as you've mentioned, was not an issue because we listened to almost everybody. We had children and their parents, as well as every other person in the community. So there were different age groups. And if you're talking about age and maybe it's just children you're talking about, I don't know which age group specifically, but if it's the children, we had children of all ages. Praziquantel is administers to children 5 to 14 years in school. So the children we met in school, they told us if they had taken the drugs, why they did not take the drugs. And we listened to their parents as well within the community. So we didn't find age as a limit in our study.
I guess that answers it.
Thank you.
Christine. And with regards now to the set of, I would say, guidelines that within the setting, what is the next step? I believe it to try and implement that to see whether these corrective measures will, let's say, maximize the adherence then to MDA, correct?
Yes, correct. Actually, this study was carried on as part of a study, which we done. And what we realized from the field was immediately translated into action, and we had good results. The countdown study, to give an example, to say in summary, the countdown study involved in carried out in observing and carrying out mass drug administration. So at the same time, what we observed in the field, we immediately implemented it in the next distribution campaign, and we saw the results. So it's something that has been tried out, and it's something which had positive results, and we encourage it to be carried out on a wide landscape in other places.
Well, that's great. And it just lays then the basis for the final question from Anouk. Do you think that you can develop then an adaptable protocol that can be used by program then to assess reliably who is being left behind and then tailor this messaging and engagement of specific groups?
Actually, I think that will be the final part of our work in Countdown. Considering the other protocols that are being developed in other parts of the project, like that on female genital schistosomiasis in Nigeria, I think it's going to be an interesting part for Countdown to develop a protocol in order to carry out mass drug administrations in different areas specific to the context of that place. So it's not something it's done right now, but I think that's part of the plan to carry out, to produce a protocol on this. Thank you, Anouk.
Well, that's great, then. So we do look then forward then to the end of that plan. And thank you to all the speakers of this session for the fantastic then set of talk. I will now then hand over to Debbie to give directives then on the short break now.